Hi. As the name implies, metabolic regulators are used to regulate the metabolic process. In some fermentations, we need to control the fermentation so that the timing and the side products can be controlled. In this session, I am going to talk about metabolic regulators used in the fermentation. And it used to be incorporated with the media. That's why we are discussing it in this session. Metabolic regulators can be classified into three different types: precursors, inhibitors, and inducers. Precursors are the compounds which is incorporated to the product. So it will be a precursor, and the bacteria or the microorganism or the animal plant cell, whatever it may be, will be synthesizing or the modifying the precursor to get the final product. Inhibitors will inhibit the microbial products in some way. Sometimes it is used to inhibit the byproducts, or sometimes it used to inhibit the degradation of our desired product. So different ways we can use the inhibitors. Induces are the compounds which induce the production of a compound. When we are comparing precursors and precursors will be a part of the product, but induces are not. Induces will induce the production. It will induce the bacteria or the microorganism to produce desired products. So we have three different types of regulators. I'm going to discuss each of these regulators in detail. Firstly, we have the precursors. Some fermentation needs specific precursors. The requirement of precursors will vary depending on the type of fermentation. And the use of precursors is like it increases the product yield. So, and it also helps to control quantities, and it is relatively pure form we are adding. So the precursors, bacteria used to produce, the microorganism used to produce the compound from de novo, like means they are producing from the products of the Krebs cycle or the glycolysis or some other compounds. They are producing from the scratch, starting from the beginning. They are producing. When we are adding the precursors, bacteria, what they do is that they are modifying the precursor to get the desired product. So the energy requirements and a time requirement for the bacteria microorganism is less. It's also true for the plant and animal cell culture. So any culture, if you are adding precursors, the organism is will produce the product very easily. So that's the point. So maybe we will produce the main component of the or the basic structure of the compound. The organism will modify the product, the compound, to get the desired product. And we used to add it in controlled quantities. We will maybe in the particular state, if we the product formation in the stationary phase, we don't have to add the precursor in the beginning of the media. We don't have to. We can wait until the organism reach the stationary phase. So during the time of stationary phase, we can add the precursors. So the organism will modify the precursor to get the desired product. And we used to add it in relatively pure form. So most of the precursors are synthesized chemically, so we can get it in a pure form, so we can add it in pure form. It's like a fed batch process. So once we have started the fermentation, the lag phase is go over, log phase is over. Once it is in the stationary phase, we will add the precursor little by little, so the microorganism can actually modify the precursor to get the final product. So the precursor will be incorporated into the product. One of the classical example is phenyl acetic acid. Phenyl acetic acid is a precursor for the penicillin production. So in the traditional way, the penicillium is producing penicillin from its metabolic pathway. So once we add the penicillinic acid, which is the basic structure of the penicillin, the microorganism don't need to synthesize all compounds to get the penicillinic acid. It's as an intermediate of the penicillin production. So if we add the penicillinic acid, phe- phenyl acetic acid to the medium, the penicillium is very happy to modify it to make the penicillin. So we can save energy and time. So once the penicillium get the phenyl acetic acid, it will add the side chains to produce the penicillin. So the process is comparatively easy. It is less time consuming, and it consume less amount of energy. So the profit will be more. Another example is the D-threonine is used as a precursor for L-isoleucine production by Serratia. So same thing happens. We are providing an intermediate as a precursor. So the intermediate is provided ready-made. So the microorganism just need to modify it. So we will get product very easily, very fast actually. Nextly, we have the inducers or elicitors. Uh, these compounds either it act as a specific inducer compound, it induced production of a particular compound, or it is a structural analog. Sometimes the inducer get modified once product is formed. So in order to avoid that, we can also use a structural analog. But it induce product formation. It asks the bacteria to produce the required product in high quantity. So we need to incorporate it into the culture medium, or we will add it as in a fed batch process. The specific point, just what I say about the precursors, maybe in the stationary phase or the log phase, we can add the precursor to the medium. There are many examples are there. One of them is the flavonoids. For the production of flavonoids, we can add pinostrobin as a precursor, as an inducer. Once the pinostrobin is there, the cells will produce flavonoids. So it's an inducer, like pinostrobin used to see in insects and everything. So flavonoids are produced to resist this fungal infection and everything, fungal infection and type of infections to the plants. So once phenostrobin is there, cells will think that some of attack is going to happen, so it will produce flavonoids. Other classical example is amylase production. Amylase is the enzyme which used to degrade starch. So microorganisms do not produce amylase in the absence of starch. So what we do, we use starch as a inducer. Once we incorporate starch in the medium, the microorganism will produce amylase. If there is no starch in the medium, microorganisms don't produce amylase. So to increase the production of amylase, we used to add starch into the medium. And the induces are essential for the fermentation of genetically modified organism. Genetically modified organism used to have some inducers in the medium, then only it will work properly. And the IPTG, you might have heard about it. IPTG is an inducer of lacopron. So most sometime we are putting our gene of interest under the control of lac C gene. So IPTG, which is an analog of lactose, allolactose actually, it can be used as an inducer. So it is necessary for genetically modified organism. In order to control its expression, we always have an inducible promoter for the heterologous proteins. So inducers are essential for genetically modified organisms. Finally, we have inhibitors, another metabolic regulator. It is used for three different purposes. One is to redirect metabolism towards target product. So maybe there is two or three sideways in a reaction. So we want only one product. We use inhibitors to prevent this side reactions and everything. So it's every uh, compound. So the whole pathway will lead to a single reaction, or it can be used to reduce the formation of undesired intermediates. So the intermediates may end up as a byproduct, and it will uh, complicate the uh, downstream processing. So sometimes we use inhibitors to reduce this undesired intermediate, or halt a pathway to prevent further metabolism of the target products. So the inhibitors can be used to inhibit the degradation or modification of the final product. Sometimes it happens the final product what we need may be more modified by the bacteria or degraded by the microorganism. So we use the inhibitors to inhibit further metabolism of the target product. So it is used to redirect metabolic pathway to target products, reduce formation of undesired intermediates, and halt a pathway to prevent further metabolism of the target products. So different we can use inhibitors for different purposes. Uh, then the examples including sodium bisulfate, sodium bisulfate. It is used for the formulation of glycerol by Saccharomyces cerevisiae. It's an inhibitor, but it helps the Saccharomyces cerevisiae to produce more of glycerol. And genetically modified microorganisms, basically we use antibiotics in the medium. Antibiotics an inhibitor. So the use of antibiotic is that the microorganism without the particular gene won't grow in the medium. So antibiotics used as an inhibitor to make sure that the other organism without the plasmid or without the foreign gene won't be growing in the medium. Another example is bromine. Bromine inhibit chlor-tetracycline in tetracycline production. When we are producing tetracycline, we need tetracycline. But what the microorganism do, once it has the tetracycline, it may modify it into chlor-tetracycline, which is actually a byproduct which cannot be used for pharmaceutical purposes. So if we add bromine in the medium, it will prevent the formation of chlor-tetracycline. We will only get the tetracycline production. Then one other special requirement in some cases is cell permeability modifiers. As the name says, it is used to alter the permeability of cell. In the most of time, we want to increase the permeability of the cell so that the product will be intracellular product will be transported to the extracellular space. So the process of purification will be more easy in most cases. So cell permeability modifiers modify the cell wall or membrane permeability. Both are important. We need to increase the permeability of cell wall as well as the membrane depending on the product. It is promoting the release of intracellular products into the fermentation medium. As I said, the intracellular products will be transported out to the extracellular space. The use is that sometimes the intracellular product acts as an inhibitor. So if it is transported out, it won't have an inhibitory effect. Penicillins, the different types of penicillin or surfactants like SDS can be used as a cell permeability modifiers. It is generally used in amino acid fermentation to get the amino acids into the medium. It will be used in very minute quantities. If you add penicillin in huge quantities, it will kill the microorganism or other organisms. So we are using it in very small quantities so that it won't kill the organisms. It only increases the permeability of the permeable cell wall permeability. You know how the penicillin is acting. It is degrading the cell wall at right concentration. But if we are adding it in very minute quantities, it won't kill the microorganism, but it will only just increase the permeability. Surfactants, same thing. If you add SDS in high quantity, it will definitely kill the organism. So if you are using in very minute quantities, it helps to improve the cell membrane permeability. Classical example is being the glutamate production by the corynebacterium used to add penicillin and surfactants to improve permeability. So that's all for this session. Thank you so much.